Welcome to Education Wave Success Made Easy Sexual Reproduction in Flowering Plants. So, in this video, we are going to cover the topic stamen in a very detailed manner. We discussed that it has four holes, the calyx, the sepals, we know it has sepals, we call it, the corolla, or the petals, the same. Okay, and then gynoecium, the female part, and the androecium. Video, we are just going to discuss this anther and filament part. This is a structure of filament, and this filament is attached to the thalamus part. And on the top of the filament, on the terminal end, you could see these bilobed structure. This is a bilobed structure. Okay, the one lobe, two lobe. So this is why it is known as bilobed structure. And this anther is full of pollen grains. But how this anther reaches to the stage where this is full of pollen grains? Okay, so from the very early stage to the maturity, how anthers were supposed to be in the beginning, like what was the tissues or the cells lead to the formation of little pollen grains. So in this video, we are going to discuss that. So let's start. Stamen has two parts. A long cylinder stalk called filament. This is filament. And the terminal, generally bilobe structure called the anther. You could see this is a groove. This groove, when the anther is full of pollen grains, for example, this basket, if I could draw, this is a basket. It is full of apples. Okay. Now, what if I could make a little cut here? So it would be easy for me to separate the apples one by one, right? It would be easy. So it just like that, that this groove is actually will help in future in the hyacinths of the pollen grains. The pollen grains will release from here, and they will release in air, or it could be in water, or on an insect leg or wings. Okay, so it will happen in future when it will mature. So this groove is basically is going to help with the dehiscence part. But what we are actually here to learn is that, see, this anther and this filament is together will be known as stamen, and we could see that this is bilobe. Okay, so this bilobed structure, for example, if I could cut it transversely, if I could cut it transversely, this bilobed structure, if I could cut it transversely, it looks like this. See, this is a groove from where the pollens are releasing. So this whole thing is a thick bilobe structure. But one lobe carries two thecas. See, there are space between the lobes. See, so this whole anther is full of pollen grains in during maturity. So this whole theca, one, two, three, four thecas, because one lobe has two thecas. So we have two lobes, so four thecas are here. So it is very easy that this bilobe structure has two thecas. This also has two thecas. And this theca, the bilobe nature of an anther is very distinct to the transverse section of anther because it looks very different. We see from the outside that it is just bilobe. But when we cut it transversely, we could see that the one lobe is actually carrying two thecas, two spaces, where we could store the pollen grains. Okay, so this whole structure is a transverse section of stamens, which is basically asked in exams to draw it. Maybe in your board exams, it might be asked, Okay, so the anther is four-sided. So it is four-sided. It could be this structure is this one, two, three, four. So four-sided, and you could see that here are four spaces. So we call it tetragonal. Tetra means four. So tetragonal structure. It consists of four microsporangias. So these spaces are known as microsporangias. No need to get confused here. Let me discuss it again. So we saw that this whole thing was one lobe, but it has two thicas, right? We could see that it has a structure like this in a one lobe. So it is like this. This whole thing will be filled with pollen. This whole thing will be filled with pollen grains, and this is a groove. Similarly, in the second lobe too, we could see that there are two thicas. Okay. So what was this microsporangia thing we just read here? This microsporangia is the same. See, the anther is four-sided tetragonal structure. So this anther is a tetragonal structure because it has four microsporangias. It is like a bag. It is like a sack. For example, if you go to market and you want to buy vegetables, you need to have a basket or you need to have a bag, right? So it is like a bag so that we could store the pollen grains in it. So this microsporangia is similarly like this sac. We call it pollen sacs too. It is like a pollen bag, you could say. So microsporangia develop further and become pollen sacs where we could store pollen grains. This is what microsporangia is. So this microsporangia will further develop. So you could say that this microsporangia is in longitudinal because this whole uh, thing will be filled with pollen grains. This whole length of microsporangia will be filled with pollen grains. Longitudinally, this will be also, it is like a vessel of glass. So this will be filled with pollen grains. This structure, if I could draw this here, one theca and also two thecas. One, two, three, four thecas. But what? One lobe, second lobe. So bilobed but four thetas. We could see here that this sporogenous tissue, it has a tapetum. So tapetum, what is tapetum here? It acts as a nourishment tissue to the sporogenous tissues so that this could divide further and form pollen grains. So to do that process, this tissue needs energy or it could need food. So tapetum provide nourishment so that the whole pollen grain development process could occur. Okay, so tapetum is for nourishment. But what does the middle layer do? And we have this the outer layer which is known as epidermis. Then we have this endothecium layer. Okay, then we have this middle layer, and then finally we have the tapetum, and this is the connective tissue which joins the two lobes. So this is all we had to discuss here. Now NCRT has a question which I should be discussing later, but still, can you think of how tapetal cells could become binucleated? Now sometimes you will see that the tapetal cells, you know that cells have only one nucleus, but tapetal cells sometimes, or you could say that it has nucleus which is known as binucleate condition, right? That the karyokinesis means that the nucleus divides into two halves, but the cytokinesis does not occur. It means when divides, it is destined to be happen. What? That the cell also divides. But in the tapetal condition, in the tapetal cells, the condition is not similar like that. No cytokinesis occurs. So you could see this situation where both the nucleus end up in one cell. Okay, it ended up in one cell. So that's why the tapetal cells have two nucleuses. So thank you so much for watching. Please subscribe and comment and like also.